Konnichiwa, minasan. I'm your friendly neighborhood science otaku tutor, Ichiro Cyan. Yoroshiku onegaishimasu. Hey everyone, thank you for coming tonight. It's going to be an interesting night tonight, especially with something I found. So, kind of like how I've been kind of playing with here, with last stream, I'm going to be giving some science news that I found interesting this week. Some very recent, actually, and help explain that. And then I finally got the game I really want to play working, somewhat. Well, we'll see. I know at least off stream, I got it to work. Now, on stream, that's probably going to be a different story. So, actually, I want to talk real quick about something amazing that is happening. So, just yesterday, I finally put in my request to get my live 2D model done. I finally settled on an artist and a rigger, and so I can't wait until it will be done and I can show everybody and we can have a lot of fun with it. I'm really excited about it. As well as I've been reading a lot of things about tips and tricks for VTubers, especially if you're just starting out, and that has been very insightful, especially as I talk to some other people about it. Some people who are of various different levels of their VTubing journey, from just starting out like me to even more seasoned professionals that have been doing this for years. So it's been really fun and very informative. So I can't wait to see about start working the stuff in, slowly but surely. Okay, so with all that out of the way, let's go ahead and get into today's first topic related to science. So hold on here. Let me move some stuff around. Got to get my little tablet in front of me so I can draw on my chalkboard. Let's see. That's the one I want. Okay, so it's two science-related topics of interest that I want to talk about today. The first one is related to one of my personal favorite things, molecular biology. You know, how the proteins and the DNA and everything work inside the cell. So that's one of my major interests. And that can span over many, many different types of fields when it comes to biology. It can be my favorite, which is microbiology, working with bacteria, viruses, fungi, and everything like that. It can be any other type of disease where you deal with cellular processes, or in this case, cancer. And if there's one thing it's always interesting, it's cancer research. Why? Because there is so much research just coming out on it. because when you think about the fact that a lot of molecular biology techniques, especially the more later ones where we get a lot of information for it, kind of came about around the 1990s. They really haven't been around that long. And so we are still discovering new techniques and new ways to pick apart these cells and these genes and see exactly what they do and how they function. I mean, consider the fact that it was only in the 1950s whenever we figured out the structure of DNA. And it wasn't until the 60s when we figured out how that code in DNA is converted into proteins. So, yeah, this is still it's a field that's still in its infancy. So there's still a lot to learn, and that's what makes it so exciting. So, before I start into what they were talking about, the focus of the paper that I found, let's talk about cell cycle and what that is. Let's see. Oh, that's perfect. So, what is the cell cycle? Well, the cell cycle is actually how cells reproduce. And what do I mean by that? I mean that's how they divide. There we go, that's better. Now, of course, this mostly applies to what we know about mitosis, which is most people associate with cellular division. It turns out that is partially true because mitosis is not the division of the cell. It's actually the division of the nucleus inside the cell. There's actually another part that occurs called cytokinesis, where the cells finally pinch apart from the parent cell to the two new daughter cells. Now, of course, like anything in a process where the cell has to build up something, this is going to be a very energy intensive process. So do they really want to be doing it all the time? No, they do not want to be wasting all of their resources just to divide. And so that's where the cell cycle comes in. So you can imagine it like a clock. And the clock looks as follows. Between 12 o'clock and 3 o'clock here, this is what's known as the G1 phase. Now, what does G1 mean? G1 means first growth phase. So this is where the cell is preparing to divide. And as the cell is preparing to divide, it's going to need to make all of its resources. So that means we need all the proteins, all the different little phospholipids that make up the membranes. We need all the sugars. We need to build up everything, to fuel everything. So this is where the cell is growing and getting ready to divide. Then, from about 3 o'clock to around 5 o'clock is what's known as S phase. S phase, also known as the synthesis phase, is where the chromosomes actually duplicate. This is where your DNA is replicated. So that's DNA replication. Now, after that, we go from about 5 o'clock? Yeah, about 5 o'clock all the way up here to around 10 o'clock. And this is known as, let's try that a little better. I got a little tight in there. So I'm just going to write it off to the side here. G2. And the G2 phase is another preparation stage. So here we are making sure these are our final checks. These final checks are, okay, do we have enough resources in order to divide? Do we have all of our DNA in order? Has everything worked properly up until this point? Because once we get to the next phase, there is the point of no return. And what's the next phase? It's known as M phase. M phase is from 10 o'clock all the way to 11:59. Now, there's a reason I'm being very specific with that. M stands for, of course, mitosis. So this is where mitosis occurs. So this is where the nucleus finally divides. And it splits into two, and then finally we have this last phase, which is known as cytokinesis. And cytokinesis is actually where the cytoplasm finally divides. So so after that, we finally get our two daughter cells. And since it's a cycle, we can go right back into G1 to prepare to divide, go back into DNA replication, go back to final checks, go back to mitosis, split the cell across, and then continue. Why? Because it's a cycle. Now, this is what's normally taught in high school. However, what they don't teach a lot in high school is there is yet another phase. And this other phase kind of is a pull-off. You know, kind of like think about you're on a roundabout and then you have some place where you can just kind of pull off and get off it so you're not stuck in it everywhere. And that is known as G0. G0 is known as the rest phase. Let's do better. There we go. G0 is rest phase. So, fun fact. Majority of the somatic cells, so in other words, the cells that are present in the body are actually in rest phase. They do not divide. Instead, they are waiting for a signal to come to tell them to divide. And then, of course, once they get that signal to divide, they can go enter right back into G1 and start the preparations for division. And so that is the cell cycle. Now, of course, this is kind of potent. I mean, remember, this is cellular division. So there's a lot of energy here, and there is also a lot of chance to make mistakes. So what we want to do is we want to have some sort of proofreading in there. What the? Hold on. Uh-oh. That might not be good. One, that should be fine. Okay, we'll see how long it lasts. I just got the low battery warning on my iPhone I use for streaming. That's great. Anyways, so we'll just go as long as we can, and if it gives out, it gives out. Come on, low battery mode. Come on, low battery mode. Okay, so going back. So what happens is you got to have the signals that is going to tell it to go into division. But as I said, we have checkpoints. We have proofreading that occurs. And there are specific areas where those occur. They occur at the beginning, the start between G1 and S. It occurs between S and G2. And then the final one occurs between G2 and M. These are all checkpoints. Now, what's happening at the G1 checkpoint? We're trying to see, this is where we're seeing, is everything ready? Are we on our way to start to have everything ready in order to divide? If we aren't, then guess what? We do not pass go. We do not go into S. It stops right there until we are ready. The next one is the S checkpoint. At the end of the S checkpoint, this is after DNA replication. So this is where we're going to check for mutations. This is exactly where we're going to check for mutations. We're going to check to see, have the chromosomes divided properly? Is there the right number inside the to be prepared? Do we have any errors that have occurred? Because remember, DNA polymerase is not exactly 100% error proof. It makes one error every 10 million bases. So that means there is still a chance for it to mess up. And depending on where that mess up occurs, that could be the difference between a functional gene and a non-functional gene. So that's why we are looking here. And also, maybe something has happened inside the cell that has changed conditions and may have affected the proofreading, or may have changed one of the bases around that where it shouldn't be that way. And so it causes some mismatching to occur. That's what we're checking for here. And then if we find those errors, we have to fix them. We cannot proceed until all these errors have been eliminated. So then, after that, in G2, the G2 checkpoint, this is actually checking to see, do we have our final preparations ready? Are the microtubules ready? Do we have spindle fibers ready? Are there enough? Is there enough to go around? Is the cell large enough in order to divide? And everything. And then after that, mitosis, and then technically there is like another checkpoint after mitosis, the M checkpoint, which is of course to see, did mitosis occur properly? If mitosis didn't occur properly, then we can't go forward. We either A, have to fix those errors, or B, well, there's only one thing a cell will do if it can't fix its errors. And that is something known as apoptosis. And what is apoptosis? This is basically programmed cell death. In other words, if there is too many errors, it will go into a self-destruct sequence. In other words, it would rather self-destruct than carry on those errors and anything there. Why? Because we want to have a healthy cell, something that is able in order to support the organism, not a disease cell. Now, of course, the more times you go through the cell cycle, the more times you could have mutations occur. And so that's why, and whenever some cells have gone through the cycle so many times, and their DNA is getting more and more frayed, it's accumulating more and more damage and mutations in that eventually they just undergo cell death, and a new, younger cell will replace it. All right, so what governs all this? Well, of course, given that this is the body, there's only one thing that can govern this, and that is a protein. And one of the famous proteins that governs this is known as P53. P53 is the protein for the S checkpoint. So, in other words, this is the one that checks to see, is the DNA intact enough in order to divide? If not, it will send it back to fix it. And if it can't fix it, it sends it off to destruct. So, in other words, P53 is a very important protein for this. And in fact, it was one of the first of these what are known as tumor suppressor genes that was discovered. So you can tell by that name, a tumor suppressor, what this is mostly associated with. And how this was discovered is the fact that when you have a disease that has, where you've inherited 2 copies of the broken P53 gene, I'm trying to remember what the name of it is. It's not DiGeorge, it's not Fanconi. Can't remember it off the top of my head. But what happens is they saw in people who had this mutation higher incidences of all different types of cancers. And so they figured out this protein was required in order to reduce the risk of cancer, and then eventually figured out what its function was here as the checkpoint protein. So now you might be asking, well then how does cancer occur if P53 is active? Well, here's the thing. What if a mutation occurs within P53? If a mutation occurs within P53, in other words, one of those just random mutations that occurs inside the P53 gene, would that protein be effective? No. And so that's one of the ways that this starts to break down, is that if there is a mutation that occurs in P53, then that means, let's see here, I'll use purple. That means this checkpoint no longer exists. Well, if that S checkpoint no longer exists, then what's going to happen? Are we going to catch all these mutations? No, we're not going to catch these mutations. And so these mutations proceed on through and get inherited down into the daughter cells. And of course, as you increase the number of mutations, you can start breaking down more and more of these checkpoints. What I'm also not going into here is all the cell signaling that's going on. They might be getting signals to say divide, 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 divide. And so as they hear that, they're going to continue to run through the cell cycle and gathering up these little mutations, because again, it said every time there's a division, there's a chance for a mutation. And then finally, we have just a case where now the cell cycle is unregulated. And so you have unregulated cell division. So it's basically stuck in this loop, and all of its daughter cells are stuck in this loop as well. Now, of course, what do they need? They need nutrients. And cells are greedy. They're going to send out signals saying, hey, I'm hungry, feed me, feed me, feed me. Who here has seen Little Shop of Horrors? So you can think of them going like, feed me, Seymour. That's what those little cancer cells are doing. And guess what? The body is responding. And so that's how it starts sucking up all the resources, while everything else, of course, is trying to vie for those same resources. But now you have these cells that are just growing wild, taking up all the resources. And that's mostly what happens with cancer. So one of the big questions we've had is, well, you know what? Then how in the world can cancer, because cancers also have their own defensive mechanisms. That's right. They can actually have their own defensive mechanisms. Like, for example, let's go back. There we go. So let's say this is because, of course, what's going to be on the lookout for for any cancer cell? Immune cells. So, like, for example, how about I write the right letter? Natural killer cells. They go around and patrol to make sure everything is right. There we go. Everything is right with your cells to make sure everything's kosher. Well, guess what? They will be looking to see for, is this a cell that belongs to you? A self cell? Or is it non-self? And if it is non-self, it will go, all right, good job. You are supposed to be here. Wait a second, you're not. And of course, then it is going to kill it dead. Well, some cancer cells can actually masquerade themselves as a self cell and therefore evade that. Or they can also, like, say, if you have something else here, another famous immune cell, like, say, a macrophage. Macrophage, the big eaters. They're the ones that go around and go and eat all the dead debris, all the bacteria that's trying to cause infections, and all the, of course, things like cancer cells. Well, what happens is, if you have a cell, let's go back to purple here. I got some nice cancer cell, and that's definitely not it. Here we go. So we got our misshapen cell here, our cancer. And what it does is it can secrete proteins, which basically says, hey, there's no cancer cell here. Keep moving along. And they can. So we've known a lot about these cellular defenses that they have against your immune system and to protect them. But it turns out, just about 5 days ago at MIT, they found another mechanism, and it actually is a cellular mechanism. And so what is it? Remember how we said that we break P53 and it is non-functional? Well, there are actually proteins inside the cell known as chaperones. And if there's one thing about science, we always name something by its function. So with something like a chaperone, what do you think its focus is? Chaperone. Well, when you think of the word chaperone, what do you think of? There we go. A chaperone is to assist. And if you have all these proteins about, are you going to want to make sure they fold over into the right shape in order to function properly? That is correct. You are going to want to see that happen. And so that's the focus of these chaperones. In fact, the old name for these, because of the way we discovered them, are known as heat shock proteins. Heat shock proteins. And one of the famous ones is heat shock protein 90. Heat shock protein 90 is a chaperone that is found in all mammalian cells. And it helps the proteins to fold. Well, of course, if P53 is not folding properly, anytime there's something's not folding properly, it starts sending alarm signals going on saying, something ain't right in this inside my cell. If all I'm doing is sending proteins to be destroyed, something's wrong because they're not folding properly. So it turns out one thing they've noticed is that in a lot of cancer cells, these heat shock proteins are increased in expression, which means there's higher levels of them. But what would be the reason for that? To make sure proteins fold properly. And remember, what are we gathering every time we divide, especially if P53 is broken? Mutations, which means proteins aren't folding properly. So it turns out they jack up the expression of these heat shock proteins in order to make sure the proteins fold properly, including, and this is the one that amazed them, broken P53. So in other words, it maintains its shape. However, it is broken. It is not performing its function, but because it's maintaining its shape, as well as all the other proteins that might be active and supposed to be like suppressing the tumor or giving the signal to halt, or even just other functions like that, they are folding properly because of all these heat shock protein, these chaperonins telling them how to fold properly, but they are not functioning. And so it tricks the cell thinking everything is fine, whenever it turns out everything is broken. And so they called this, the title of the article, Cancer's Secret Safety Net. And now, knowing that this exists, this can tell us a bit more about what can we do in order to address this. Because we found out that just tackling drugs that target HSP90 really didn't work. And it turns out, why? Because there's so much of it present, we can't drug it all. I mean, before it starts affecting other cells that aren't cancerous, just the normal cells themselves. So, yes, if you want to read more about this, and I always highly encourage that, this is, I found this on MIT Chemistry, on their X, Twitter, whatever you want to call it, page. And yeah, it's literally called Cancer's Safety Net. The research for this just published like 5 days ago. Definitely go and check it out if that is something that interests you. Because I know that's definitely something that interests me. All right. So that was the first topic I wanted to talk about today before we see anybody coming. No, not yet. All right, cool. We'll move on to the second topic. Let me just clear the board here. Actually, I'll just do it this way. It's more fun. Eraser. Big eraser. Just go through everything here. make sure we got that. Perfect. Yes. Okay, so I always kind of want to have at least two topics to talk about. And so this is the other topic I found earlier today. And it was interesting because tell me there is nothing more interesting if you're than the human brain itself. Let's see here. Let's go back to chalk. So yeah. So talking about the human brain, a little bit of psychology. Is it one of my fortes? No. Do I know enough to be dangerous? Yes. um. So, and it turned out it was a study that was done in 2023 at Stanford University. And it was on the effect of stress and negative thoughts on your mood and your emotions. And this fun term for the psychologists and those interested, neuroplasticity. Neuroplasticity. What does that mean? Well, again, let's break it down. So, neuro, well, that means it's going to be related to the nerves. And plastic, well, if something is plastic, what does it make you think of? It makes you think of it being flexible. So, put it together. What does it mean? Flexible nerves. But, well, I mean, nerves are flexible if you think about it. I mean, they're kind of floppy and everything. So, isn't that what we think of as flexible? Well, in terms of this, we're actually talking about how they can rewire themselves. So they can rewire the pathways. So we're rewiring the paths inside the brain. And not even just inside the brain. You can find neuroplasticity in the peripheral nerves as well. Where if, say, one pathway is blocked and it's a pathway that's commonly used, the body will find a way to bypass it and go around it. And that's what neuroplasticity is. And of course, this is a big topic. Why is this a big topic? Well, a lot of common disorders that you can think about come up when you start talking about neuroplasticity. And I'm not talking about neurological disorders. I'm talking about psychiatric disorders. Because stress, autism. In fact, one of the big beliefs with autism is it's a breakdown in the pathway that prunes back all the different pathways that build up in the young infant brain. And so they believe that, well, we have all these different pathways, and that's what causes a lot of the confusion in the messages that are being sent in the brain, and that's what causes a lot of the sensory overload, because you have, like, say, only one thing that's supposed to have maybe 3 paths, but yet it would go into 100 other paths, which is exciting all these other nerves, which is causing issues. Would Asperger's be included in this? Yes, Asperger's would be included with it, since the old term for Asperger's was is a form of autism. They used to call it mild autism. Now Asperger's has been loaded into autistic spectrum disorder, ASD. So you can see why this is a big topic, especially because All right, show of hands. How many people get stressed out at work? Yeah, everybody's hand should be up. I can't think of anybody who has ever had a really good day at constantly a really good day at work, and if they say they are lying. There's always at least one day where even if it's a job we enjoy, it drives us absolutely insane and stresses us out. And well, so the interest in this study was the fact that they were looking to see if it was possible that by constantly talking about our stress and perseverating on it, could we be actually rewiring our brains to be more sensitive to stress? In other words, to beat that, because of that, that leads to a rewiring to be increasing things like, say, cases of depression. Now you can see why this is a very interesting topic. But there's a catch to all this. And the catch is this. That doesn't work. Let's try a different color. There we go. I did research into this because, again, I mean, any scientist is going to do their due diligence on this, because this is major. If this is true, and especially if this was discovered back in 2023 at Stanford University, of all places, I mean, come on, that place is known for its, Stanford medicine is well known. Then that means there should be an article out there. Oh, that sounded great. And it turns out this research doesn't exist. Everything I just told you is actually a lie. But where I found this was in a Twitter post earlier today. And I shared it. And I shared it with some friends because, again, this is amazing. But it turns out, and this is the post. Let me bring it up more here. This is the post it was. It says, research shows repeated complaining physically rewires your brain to prioritize stress and negativity. The way we speak about our daily challenges does more than just frustration. It physically alters the architecture of the brain. Again, that catches your attention. Is that why I mentioned it sounds like how drug addiction works? Interesting. We'll come back to that later. But yeah, I mean, this catches your attention. I mean, here, think about this. Again, how many times do we deal with stress? And we know a lot of stress causes depression issues and anxiety and all these disorders. But when I looked into it, I went to the University of Stanford site. I went to their actual site to see, does this exist? And whenever I looked, the answer was no. And I even did a Google search on it. Oh, that's right. Window. Stuff. And I looked up the exact article that was given, the exact title of it, and at Stanford Med. And it turns out it does not exist. let me check here. Let me see if I can check it on this screen to see if I can see it. Get it to you. Yeah, here we go. But yeah, you can see here. This I searched for it. Stanford University. The article doesn't exist. I went and checked Google Scholar. Google Scholar does not have anything on this. And mind you, Google Scholar is a great resource for looking up information and access to articles that are out of scientific magazines and peer-reviewed journals. So then I went to on PubMed, where we have all the journals. It's so easy for people to spread misinformation about biology. It's so complex, many people have no good way to fact check. That is correct. That is so correct. And look here, I filtered it for Stanford, the publication date, and the title in it for neuroplasticity or neural plasticity. And this is the only article that comes up, and it's not even related. So in other words, everything that I just said, I mean, are there actual studies on it? Yes, actually. There was this was an editorial back in 2017 in actually a journal called Neuroplasticity, where they are looking at stress-induced neuroplasticity and mental disorders. But look here, it's 2017, not 2023. And it's not Stanford University, it's Texas A&M and Baylor and a few universities in China. So there is studies on it, but guess what? But it's not Stanford University. So what was it then? Well, if we take a really close look at it, I'll tell you what I think it is. I think this is something that someone came up with everybody's favorite boogeyman nowadays. AI. There have been cases of where people will just throw whatever they want into ChatGPT, Grok, Gemini, what's the other one? Anthropic's, Claude. All these different chatbots, and they will go and give them exactly what they tell them they want. It'll give you exactly what you tell it wants. But the thing is, and people have used these to write papers. There was a famous one where lawyers were using it to write up their legal briefs in court and submitting them to the court. And they found, and one thing it can do is it can come up with references that doesn't exist, studies that don't exist, references that don't exist. But because, again, I'm being honest. There have been many students in college that's one way they got caught using AI is the fact that the studies they quoted, they referenced don't exist. And I'm trying to remember. I forget what legal firm it was, but they literally had to put out a message to, do you have a source for these court cases? Not on me currently. This happened, I think, like last year or the year before, I think. Like, whenever everybody wants to use AI to do everything. I have to dig it up and find it because I remember hearing about it. Actually, hold on. Google Food to the rescue. Yeah, that's the the one that's currently here. Let's see here. Court case. What I say? Court. Legal briefs. That's it. Legal briefs. AI warning. Did it happen? Here it was, AP News. It was last year, actually, October of last year. Found it. Here it is. Later. Mistake-filled legal briefs show the limits of relying on AI tools at work. AI has no actual intelligence in the very simplest possible terms. It is a predictive text generator with an AI algorithm trained to give relevant answers to all requests. That is correct, Ellie. In fact, a lot of us who are big into AI, me included. I mean, I am an AI if you think about it. Ooh. We don't even call it AI. We call it by a different name that we've called it for decades. Machine learning. Because basically, whatever goes in is whatever comes out. Computers cannot generalize well. They cannot theorize. That just doesn't happen with computers. I mean, that happens with human brains, but not with computers. And that's one of the ways we gauge intelligence and things like this. Some things that need a bit more than just brute force. This is what it is. Thank you very much. But because it is the big thing, everybody's worried about AI this, AI that. It's corporate buzzwords. If you really want some good resources on talking about that, especially when it comes to the difference between what they want to think AI is and what it actually is, there was a great Linus Tech Tips video that he did back in, I think it was the beginning of last year, where he talks about AI and really sets the record straight on what AI is and what it is not, and a lot of what people think it is, and it is not. But then you have, and if you really want to see a lot about the history of it, and I highly, highly recommend this. It is actually one of my favorite YouTube channels, and I will promote this like crazy, is Welch Labs. Yeah, Welch Labs. He studies a lot of stuff dealing with AI models and even about the history of AI in this one thing called video series. It's called Learning to See, where he actually goes through all of the beginning from the very start of AI and all the way up to, well, what was present day at that time. Let's see, this is 9 years old, where he walks you through even teaching a computer how to count fingers and running it through himself. I highly recommend it if you're interested in this type of stuff. This is one thing that got me really interested in it. And he has even another series on neural networking, which is even better. Back in 1992, the Adams Family pinball machine had a machine learning algorithm that let it flip a ball magically on its own. Oh, that is so cool. That is so cool. I will have to check that out. But yeah, no, as I tell a lot of people, I am one of AI's biggest proponents, but I am also one of AI's biggest skeptics. Why? Because, again, if it's used properly in the way it's supposed to be used, like say here, learning how to see, computer vision. This is exactly how those self-driving algorithms and the Teslas and everything work, is by being able to see what it is in front of them and it's around them. But then, or in my own personal interests, there was a group that took the large language model. So in other words, the same type of AI algorithm that GPT is, and all the other big ones that everybody thinks about, and used it in order to, instead of trying to predict like what to write or what to think about, predict protein structure. Protein structure predictions is one of the biggest holy grails of computational chemistry and molecular biology, because there's a lot of proteins we know exist and we don't know what they look like. Why? They are hard to isolate, they are hard to crystallize and purify and get them into a state where we could be able to, well, nowadays, throw it in an NMR machine and do it. Or back in the old days, whenever they first determined the first protein structure, which would be Max Prout's in, I want to say it was early 1950s. Yeah, it was the early 1950s. The first protein structure to be discovered was myoglobin, which is the oxygen-carrying protein inside your muscles. That was the very first protein to be structure to be discovered. How? X-ray crystallography, shooting a bunch of X-rays at protein crystals. And mind you, they've been doing this for decades in the case of crystals, crystallography, mineralogy, and things like that, in order to see the structure of the crystals inside. And they went, hey, well, let's apply this to biological molecules. Let's try it with a protein. Yeah, it turns out a lot of them don't like to become crystals. A lot of them are amorphous. In other words, they have no shape to them, and they form amorphous powders, and you cannot find out, you can't use X-rays to find that out in X-ray crystallography. So that has been a big deterrent in figuring out protein structures. Now, with the advent of solid-state NMR and solution NMR, where we are able to look more into solution structures and see what they actually look like in their natural habitat, per se, their natural environment, as well as AFM, atomic force microscopy. That's been a really eventful one for that too, where we use a very, very thin needle to transfer over top, to kind of trace over top of the proteins in the cells. Or any molecule, to be honest. The needle is so fine, it will just trace right over every single molecule, every single atom of every single molecule present. So we really don't have a good idea of what a lot of these proteins look like. We have their sequences. That's easy. That has been around since those methods have been around since the Sanger sequencing. Well, no, Sanger is DNA. Edmund degradations. How old is Edmund degradation? Edmund. Degradation. It's not Edaman. It's Edmund. There it is. Edmund degradation. Here we go. Yes. When did Edmund degradation come about? That's the process. That doesn't tell me anything about whenever, how long it's been around. After 75 years, okay, all right. So that here, 75 years in 2025. So yeah, 1950. I was right, and that was just mainly a guess because because that's usually about where most of the molecular biology techniques kind of came about. We're in the 1950s. Anyways, so that's easy to figure out, but to figure out the actual structure, that has been very difficult. And um, so having it where we be able to predict it, I mean, Google actually has a hand in this. The AlphaFold, which uses the Google DeepMind computer and algorithms and AI algorithms and things like that. AlphaFold has been really good at helping determine some protein structures. But the one that I like, that I found out about, was the one that uses the ChatGPT algorithm, but instead of training it on text, trained it on protein structures. And that we know, okay, well, we know that if this type of sequence, so we can figure out, okay, well, if it has this type of sequence, and it's most likely to come into this type of a structure. And for us to be able to predict that easier. Of course, there's always stuff coming out about AI helping to assist in figuring out chemical syntheses. I have done many a retrosynthetic pathway before. And mind you, retrosynthesis is fun, but then I'm crazy. If you don't know what retrosynthesis is, it's basically when you're in organic synthesis, you synthesize in order to build up. And in retrosynthesis, you go in the opposite direction. So let's say, for example, here we go, good old classic benzene ring here. Let's say you are looking at this one, aniline. And you have to describe, well, how did you get to aniline? How can you make aniline from something that's easier to get a hold of? Like, say, you're trying to produce this on a wide scale, and you want to use some cheap precursor. Well, you can think, okay, well, what kind of things do I know that will give me this aniline group and give it to me easy? That is accessible. Well, I know I can make it from nitrobenzene. So I know I can reduce it from nitrobenzene. And then, well, but I can go further back and get something that's even more easier to get. And what is that? Well, I know how to make nitrobenzene. I just need to go start with benzene, and benzene is a solvent that's very common. So that's what retrosynthesis is. That's a simple version. When you get into natural products, that can be very, very, very complex. And we're talking like 88 steps. And then you figure out, well, that didn't work, so now I got to try this. And that didn't work, so now we're going to try this. And then, oh, we had to develop a whole new style of chemistry procedures in order to get to this. And then that gets used in another process and so it just builds up on itself. Wouldn't it be nice to have something to help you kind of get a start on it? So that's where some AI research in chemistry is. Or we have all this data coming in about different medical diseases and how the cells work. Wouldn't it be nice to have a way to sift through everything and all this data and put it together? And because you're interested in, say, targeting a A cell signaling molecule that is found in a lot of cancers. Yeah, that would be it. That would be nice. But you have just all this stuff there and all these different things that interact with each other. And so that's one thing that AI is being used for in drug design and helping in that. So yeah, I mean, there's a lot of useful for these AI algorithms and things that they make it with. It's just people don't use it properly or the way it should be used. So yeah. Do we know when who replaced the term machine learning with AI? Was it done to invoke the culture zeitgeist of science fiction like data, ultron, HAL, the Transformers, etc.? That is a really good question, Random. Don't know exactly who did it, but I know when it did happen. It happened around, it was early in the 2020s. It was really early in the 2020s whenever ChatGPT came out. And so suddenly, instead of having it called machine learning or, oh, I'm just going to search up there and give you what you want to hear because this is what you're interested in, people started advertising it as, this is AI. You can ask it anything. It will give you every answer in the world. Wouldn't that be nice to have every answer in the world at your fingertips? I mean, think about it. Siri, hey Google, all that has been just machine learning, but yet machine learning doesn't sound cool. AI is futuristic. So if I'm marketing it, what am I going to use? Am I going to use something that sounds actually right and technical like machine learning? Or am I going to use something that people are going to think about like AI? Because everybody's heard about AI. In fact, that's why some people have even changed the actual name of what we consider actual artificial intelligence to now a GI, artificial generalized intelligence. That's what we think of in like data, ultron, HAL, and things like that, where it is a computer, but it is able to perceive. So yeah, again, I will go back, and if you really want to hear more about that, the Linus Tech Tips video on what is AI, highly recommend that to find out more about that. All right. So let's see here. Awesome. Yeah, I mean, remember, my big goal is to make sure that I'm helping people, not tell people what to think or what to do or anything, but just help people get more knowledge and then to understand what's actually going on. Because, I mean, think about it. The main thing that makes people fear is the fear of the unknown. And so that's why education is so important. Because education allows us to understand and go further. So that, okay, we understand why this is happening or how this could happen. I'll try not to get preachy. It's hard when it's something that you are really, really passionate about. But I don't want to do any of that type of stuff. Nah, this is supposed to be fun. Yay! Um, anyways, so yeah, I thought that was all kind of interesting science bits that I found today in order to talk about. Mm. All right. Let's see here. Nice. Checking the chat logs. Nope, I don't see. Uh-oh. It's now down to 10% battery. Let's see. It was 10%. Let's see, it was 20% about, let's see, it's an hour and 18. About 30 minutes ago, it was 8. Decisions. Do I go ahead and, you know, play with fire and see how long until the battery clunks out? Or do I go ahead and start wrapping things up so that it's not going to be like all of a sudden my stream just dies out? Well, it wouldn't be my stream dying out. It would definitely be my model dying out. You'd probably hear me talk or see what that, but there would be nothing happening because there'd be getting no data to it. I can't tell you how many times I've seen that with some of my favorite VTubers I like to watch. Some of them who inspired me to do this, where you can tell whenever the iPhone battery or the iPad battery died on them, and they're just kind of standing there. Let's see. Do we play with fire and try that? You know what? That will make me very, very angry. I can tell you right now, if that dies in the middle of my game that I want to play. Because if I remember correctly, the start of this game, it is not short. What game? Heaven Burns Red. I really want to do it. And I really want to have all the reactions and everything to it. Why? Because it's Jun Maeda. It's Jun Maeda. And no, I am not afraid to cry on stream. If I really want to play it safe, could do more Hollow Cure. Because that one, it takes only 20 minutes to run through one run of it. If I really want to play it safe and play a game. Or again, if I really, really, really want to play it safe, I can just go ahead and wrap it up here. Check something real quick. Alright, I've had some better model stuff. Sorry if that's a bit off topic. I'm gonna try. Oh no, that's fine. Okay, so no, that's actually a really good question there, Mine Rat. So whenever I was doing all this stuff that was related to preparing for VTubing and all the research into it, because of course I'm a scientist, I'm going to research it to death. I found out that, yeah, like you can use, and I even tried it myself, webcams, but the problem is webcams just don't have the right technology in order to capture all the movement. So they can approximate, like say, they can be able to get like the very, what we would say, gross motion. So in other words, the very gross motors type stuff, like say going back and forth and things like that. but the more finer stuff, they can't. Not to mention the stuff that we run with a webcam has to run on your poor GPU. So in other words, if you don't have a good enough GPU in order to do it, you could be really having some issues with it. The reason why iPhones and iPads is because Face ID. Yeah, it is Face ID. Where you can just kind of look at your phone and unlock it or things like that is actually what makes it possible. iPhone, Apple really did a lot of research into like a LiDAR to be able to track the motion of your face and the shape of it and everything there. And so your popular programs like VTube Studio, Warudo, VC Face, and those can actually interact with what is known as Apple's AR kit, which is in their iPhones only after iPhone 10. Whenever Face ID and iPad Pros, regular iPad won't work. It has to be the Pro. And so what happens is because they utilize that, they can get the finer details. A lot of people do start out if they don't have an iPhone. Webcam stuff does work. It's just very rough. And since I had been planning this and working on this stuff for about 2 years at this point now, with all the planning and that, I was like, you know what? They say you don't really have to start with an iPhone, but I'm like, I'm going to invest in it, because if there's one thing I think the most important thing to invest in, it's the technology. All this stuff here, to be honest, aside from my model and the stream alert type stuff, I made all this. Yeah, I'm the one that made the background. I'm the one that made the backgrounds. I'm the one that made the boxes, the little marquee stream and stuff like that. I mean, to be honest, it just kind of depends on what you want to do. If you just want to do kind of just very basic stuff there, like PNG tubing, stuff like that there, you're just starting out, you don't need an iPhone. But if you want to have kind of a lot of the more accessibility and stuff like that, or preparing for the future, which I'm all about future proofing, which is why I invested all in the tech side first rather than investing in everything else. iPhone is the way to go. I mean, the phone I use right now as my model cam is an old iPhone 12. I found it. It's a refurbished iPhone 12 that I bought at Micro Center. At my local Micro Center. So, and it was like about $200. I have a webcam that is about $80 I bought for whenever had to stream classes during COVID. So when you think about it, not much more. So instead of, so I'm like, you know what? I'll just set aside the money and get it. But no, I mean, if you really, you can just start out with just a webcam, and go from there. It's just kind of up to you. So I know kind of my big goal and everything I want, so that's why I'm like, yeah, I'm willing to, and I know the money is going to go into it. Let's see here. Missed one here. Here we go. Random. I'd love to get some more Maeda in my life. I'm so glad he recovered from the abuse after his last surgery. Oh, you're talking about the day I became a god. Sure, Mine Rat, anytime. I mean, I said, I'm just starting out here, so I'm not going to claim myself to be an expert, but I will claim to help regurgitate what I have found out in my research. But going back there to random, oh yeah, I remember the day I became a god. Yeah, that was rough. And Jun Maeda himself is pretty tough too. Uh, no doubt, as I said in my first stream, I love Jun Maeda. Jun Maeda is one of my big heroes. His stories, I mean, of course, he's very humble, and he says that he didn't do it himself. He learned from his sensei, from his senpai. That's the name I'm looking for, senpai, who was Naoki Hisaya, and he is a hell of a writer too. Naoki Hisaya wrote half of Kanon. And then one of my favorite series that he did was an original series called Sola. It was written by him, and you could see kind of the same structure and format that you see in, let's see here. Maeda wrote Makoto and Mai paths in Kanon. Naoki Hisaya wrote everybody else, so that would be, of course, the most famous one, Ayu. Oh, yes. Oh, Soul has so much of the feels. I love it. But yeah, and so, but I also have one of Maeda's original mangas, and it got a very short release over here. It was called Hibiki's Magic. And in it, he actually talked about himself a little bit. He actually came forward and gave us a little bit of information about himself. The mangaka, like the manga artist, he was the writer. The artist was Rei Izumi. She's probably most famous for doing the Dot Hack Legend of the Twilight mangas, right? Yeah, Legend of the Twilight. And also she did some Kanon and Air mangas as well, or little short stories for them, because I actually had the Kanon and Air collection that she made. And gave us a bit of information about him and how he struggles with depression. And so that's why he likes listening to music, and how he feels music and how it applies in his life. Maeda didn't write Ayu. That is correct. Maeda oversaw the production of Kanon, but he didn't write all the stories. He only wrote Mai and Makoto's paths. The rest of them was Naoki Hisaya. So he was overseeing the general stories, so he was involved. It was his baby. It was partly his baby, but he wasn't the main writer for those ones. And so, yeah, he struggles with depression. And so that's why he first left, of course, my favorite girl, I thought it was more even split. Nope. Nope, it wasn't. um, but instead, he actually it was after Clannad that he stopped writing for visual novels. He said, "I'm done with writing visual novels." Why? Well, think about what Key's known for: the crying game. He's like, I don't have a sad story to say. And so that's why a producer from Aniplex approached him and said, "Hey, how about you make an anime or an anime? You want to make an anime?" He goes, "Sure. You're not going to want me to make it a sad story, right? Like, you can make it any story you want." He's like, "Okay, good, because I don't want to do a sad story." Little Busters was his actual last game. Mm-hmm. But, and so that's how, and he's like, "I want to do a comedy. I want to do a comedy with action in it." And the director's like, or the producer was like, "I got you. I know who to, I have some people in mind who can help you with that." And so he got with the director, Seiji Kishi. And together they put, and that's what became Angel Beats. It was around Clannad when he started thinking about, correct. Yes, around Clannad, the end of Clannad is whenever he thought it was time to move on. And of course, Angel Beats became a major hit. And then after Angel Beats is whenever he came back and created what he considers his magnus opus, which is Little Busters. It was after Little Buster, he felt like he peaked with his, Angel Beats is a comedy. It's a comedy action. And then you just have Jun Maeda at the helm, so you know you're not coming out with a dry face. But he I still need to play Little Busters. Still need to watch Little Busters too. And he said, he thought, "I peaked. This is my, I told the story I wanted to tell. I did what I want. This was and so he left being a writer for visual novels and games and just went into his other side that he does at Key, which is music production. He and Shinji Orito basically were the main big music producers at the start of Key. So a lot of the songs of Kanon and Air are done by either Jun Maeda or Shinji Orito. You can find a few other composers in there, but mostly them. And so after then they asked him, do another anime. And that's what became Charlotte. And then of course he's not doing any more work at Key, and so it was actually the next person to do the next big project was not his protege that he was training, but actually the artist who did all the designs for Key, Itaru Hinoue. And so she created the project Rewire. And that's whenever she actually got a lot of guests, writers to come in and and write for it, which is why you got. I can't think of his name. I can see all of his. Romeo Tanaka, that's his name. Romeo Tanaka wrote a lot of rewrite. He's famous for, trying to remember. Let me double check this before I say it. I always like to say I like to give right information, not the wrong information. And if I'm not sure, I'd rather look it up and give the wrong thing. There we go. Romeo Tanaka. Here we go. I know one of his major series, which got turned. I was right. One of his major series that got turned into an anime was Humanity Has Declined, which was his light novel series. But as far as visual novels, Drug That Makes You Dream, Cross Channel, Family Project, very famous visual novel writer. And so one of her favorites, so she said, "Hey, can you come and work on my game?" And of course, they got him to do it. And then of course, another famous person came and worked on it too. I would really love to see what parts he worked on, because I don't know, would you be able to tell? His style's kind of unique and easy to pick out. Ryokishi Zero Nana. Let's see, translated, his old name, Dragon Knight 07. The main person behind 7th expansion, Higurashi. I know he wrote, I think it was One Path in Rewrite. And I know his style. I know Romeo Tanaka's style. So I wonder how well did that actually mesh together. Romeo Tanaka is very, I find him kind of dry. Well, Ryokishi Zaranana likes to go mind twists. Lots and lots of them. And dark. So it'd be interesting to see, how do you have these two polar opposites come together? But yeah, and then not long after that, of course, was Maeda did his next thing was Day I Became a God, and we know how that went, and how well that went online too, to where he took himself offline yet again. That was the second time. The first time was before Angel Beats. Yeah, it was before Angel Beats. I think it was after Clannad. It was the backlash from Clannad, because Clannad was the first all-ages game to come out of Key. Don't quote me on that, but I do know it was the second time. Yeah, because he came back around Little Buster, or around Angel Beats, to promote Angel Beats. Yeah, I think it was after Clannad then. That's when he went dark online the first time. And then he went dark again. But that's whenever he then found a new calling there. He said, I want to do a mobile game. I want to write a mobile game now. And so that's eventually what became Heaven Burns Red. And luckily last year, we got it over here. So yeah. Mm. Well, the title of the stream I have running up there, saying, the marquee just saying "just yapping" sounds about right. All I've done this, all I've done for tonight. uh, how's the phone holding up? Let's actually check it. On might go dark here for a second. Oof. 5%. Yeah, I think we're going to call it here before I go completely still and the phone forces me to end the stream. Let's see here. Hour and 42 minutes. Not bad. Kind of averaging around 2 hours. Yeah, it's about what my average has been overall. Trying to build up that stream endurance, trying to build it up. Yeah, next time, make sure I keep that phone charged up better. Well, when you think about it, it's been connected to the computer, and the computer hasn't been on. Why? It's called an electric bill. When you have something that's running about 800 watts of power, and not just one, two, because I have two computers, my gaming computer and my streaming computer. That's right, dual PC setup. Yeah. I don't want to see my electric bill go sky high this month. Maybe I can put it as a tax write-off. Electric bill. Why? Because this stuff takes a lot of energy. All right. Yeah, I'm gonna call here. All right. So everyone, I want to thank you all for coming out tonight. I want you all to have a great night, and I will see you all again next week. All right. Johnny!